So everyone, we are now live on YouTube. Now we are going to start since most of us are here already. I really hope that everyone would have a stable connection because that's, I think, having a well-established connection is one primary needs. Yeah, nowadays. So. mm who's that, Miss Lita? Once again, Clarence. Siapa?
Clarence.
Oh yeah, Clarence. Yeah. So he hasn't got any clearance to enter this. So hope Mr. Clarence also able to join as soon as possible, and God give him this awesome connection. Okay, everyone. Welcome again. See you. Good morning. And I'm looking at the waiting room, and here comes Mr. Clarence again. Yeah, we are going to talk about a topic. Where the topic is gonna be introduction to microscope. I'm quite sure, everyone, that observing how the progress of the vaccination in our country has been good so far. More and more people in Indonesia got vaccinated, including you guys. Yeah, this is so awesome. This is very, very awesome. I mean, that will enable us to soon go to school. Yeah, I hope you have the same prayer with me that very soon we would be able to go to school because, yeah, the students, your older brother and sisters in grade 8, they their generation is the one which hasn't been to their own school. Yeah, one whole year they did not have the chance to go to school. Yeah, so with this progress in the vaccination process, with the this progress in achieving what's so called the herd immunity. Herd immunity means the immunity of the herd. You know what herd is, right? Herd bahasa Indonesianya itu kawanan sebetulnya. Herd itu bahasa Indonesianya adalah kawanan, kurang lebih seperti kawanan singa gitu kan ya. Disebutnya pride atau herd of, oh ya, kawanan kambing gitu kan ya, itu herd. Tapi kalau di manusia itu dipakai juga sebagai herd, berarti kumpulan manusia, lebih ke populasi. Ya, population in Indonesia. Herd immunity means this population, this collection of individuals, okay, as one whole group, this herd is immune to COVID-19. Okay, that's what actually we expect to to happen very, very soon, as soon as possible. We really need to be able to transform COVID-19 pandemic into COVID-19 endemic. Well, these are difficult, slightly difficult terms for you, maybe, yeah. But endemic means something, a condition in which something, a disease, is there in among the people. The disease is among there. It's just like stay hidden or being low profile, not showing themselves gitu ya. But it's there. But the disease has turned into something which is not threatening, not dangerous anymore. That's what we expect to happen. Okay, with the vaccination we already already had. I really hope that we can go to school. And at school, I think, this is what I think, that you will be excited if you go to biology lab. That's what I expect to happen. That's what I hope. I hope that you are excited to go to lab and to our lab and do a lot of things there. Okay, and we can have fun there. And but even though we haven't arrived at that point, I am still going to introduce you with the things, the stuff that we have in our biology lab. Okay, so today we are going to talk about this topic, the tool that we use in our lab called the microscope. Okay, it is microscope that we are going to talk about. I am going to share my screen now, and I have to optimize the sound. So here it comes, PowerPoint. Oh, here it comes. Guys, introduction to microscope. This is our topic today, okay? This is our topic today. Here on the cover of the PowerPoint file of mine, you can see that there is an image of a microscope, and the microscope, what you see on your screen is the one that we also have. Okay, we have this kind of microscope, almost exactly like this one, almost exactly like this one. But FYI, if you don't know, because I know that you know much more than I think you know, if you understand what I mean. I know that you know much more than what I think you know. um, so I just to remind you that there is not only one kind of microscope on Earth. This is not the only microscope we can find on Earth. And actually, this is not the most advanced one. Okay, this is not the most advanced one. This is the one which included into what we call the light microscope. Okay, the light microscope. And um, this one, pay attention, look carefully. This one got this part, one of this part. Okay, one of this part, because you will see something else later. Only one. This is what we call the eyepiece. Okay, and then it uses, since this is a microscope which included into the light microscope, it requires light to be able to observe things. So the light has to be provided whenever this microscope is being used. How this one would be able to provide us with light? By using the screen. The what? Not screen. Mirror. Oh my gosh, mirror. Yeah, the mirror will reflect the light from the environment, from outside external environment, maybe from the sun, from the lamp, and it will be the light will be directed through the specimen, the object. Okay, and that enables us to see the object. Without the light, we can see nothing. Okay, now I want you to go to the next slide. Here it is. Okay, this is the this is an image which shows us the two kinds of microscope. Okay, two kinds of microscope. The one on the left is the one quite similar with this one. Okay, this one is quite similar with this one. But guys, is there any of you can tell the difference between this one? Don't look at the one on the right, okay? Not the one on the right. Only the one on the left, this one, and here, this one. What's the difference between this one and this one on the left, guys? That you notice. Anybody can tell?
The shape, Mister.
The shape, Mr. Jaden. Okay, what do you mean by okay? The shape. This one is like curved like that, right? While this one is more like not as curved like that, Mr. Jaden.
Yes, Mister.
Okay, thank you very much, Mr. Jaden. Who else? What's the difference? What difference can you notice from out of this one and this one? This one and this one on the left. What's the difference? This one And this one on the left. This
The
one.
space for seeing.
Sorry, Mr. William.
The space for see the
Oh,
object.
this one, yeah. This one, right?
Yes.
Yes, thank you, Mr. William. This is what we call again the eyepiece. Okay, this one got two eyepiece. This one only got one. Good, Mr. William. Good observation. This one only got one. This one only got two. Okay, that's why this one one is called the monocular microscope. Monocular, because mono means satu, hiji nya, hiji, eta mono nya. Ocular from the word oculus means eyes, satu mata. Berarti yang pakai mikroskop ini adalah Cyclops. Enggak, Ketang. Saya juga pakai. Anak SMP kelas 1 juga pakai ya. So this is monocular, while this one is binocular microscope. Thank you, Mr. William. This is awesome. Guys, actually there is one more, at least, difference between this and this. This one on the left, this one on the this one. What's the difference?
Mister, kenapa
Yes.
kepotong ya?
This
Iya,
one?
saya kepotong.
Oh, really?
Iya, ada apa ini? Ngebengok gitu.
Wow. How about the others? How about the others? Is there anybody else got the same problem with Mr. Ethan?
Me too.
Ada
Okay.
bar, sir.
Everyone?
The zoom bar is appear.
Oh, wait. I'm stop sharing and let me re-share my screen, and hopefully everything is going to be all right. How about now?
Masih Mister.
Oh, really? Oh boy. Oh boy. This is not good. Wait. What if I show you my screen? Okay. How about now? Is it
Yeah,
still
it's already good.
already good? Oh, thank
Oh,
God. Better. Thank you. Maybe it's the network. It happened before, yeah, Miss Rita, yeah, in the webinar, right? With one speaker whose image is also like got disturbed by this situation. So it's a good thing, guys, that you tell me immediately what happened. Okay, thank you very much. Remember to always do that. Okay, now the difference between this one and this one, I mean the one on the left, not on the right, is that these are light microscopes, right? This one is monocular microscope. This one is binocular microscope. Monocular, binocular. Okay. And the difference is, look, since they are light microscope, they need light. Okay, they need light. And to provide light, this one got this mirror on the wall. Who's the most handsome of them all? So mirror, you got the mirror. While this one, instead of having a mirror, this one got this. Okay, a bulb, a lamp. So the user need to do is just to hit the button, and then the lamp will be turned on, and there will be no problem in observing things using this one, because it is something embedded, yeah, the lamp. So those are two differences. Okay, binocular, monocular, lamp, mirror. Do we have the one with mirror? Yes, we got many of them. Do we have the one with the lamp and binocular? Yes, we also have it, but unfortunately, the binocular ones belong to the different program in our school. Unfortunately, it belongs to the DCP classes. Okay, but sometimes we will try it. Okay, don't tell anyone. Okay, now the one on the left is the light microscope. Okay, the one on the right is the electron microscope. Is there any of you know or have a prior knowledge about the difference between the two, everyone? Anybody can tell the difference between the light microscope and electron microscope? Anyone? In the previous class, there was one student, one friend of yours, who's awesomely able to explain the difference. That's awesome. Is there anyone in this class?
I think from the electron microscope is
Uh-huh.
to see
Is what, sorry?
atom ya.
Pardon, sorry?
The electron microscope is more advanced. It can see the bacteria and the atom things.
Oh, okay. So the electron microscope is much more advanced, right? It could see much smaller items, objects, right?
Yeah.
Who's this? Sorry, I cannot see your name.
Ethan.
Mr. Ethan. Okay. Thank you, Mr. Ethan. That was awesome. So Mr. Ethan was right. And I know there are still some of you who already know the difference, okay? Like Mr. Ethan said, the electron microscope, the one on the right, is much more advanced. You see, the image doesn't show you the real factual size comparison between the two, because the light microscope on the left is small enough, even though this one is quite heavy. It's maybe about, it could be like 10 kilogram or something, the one on the left. But can I lift the one on the right, the electron microscope? No, because it got this table, okay, with drawers, with whatever inside, and it got these devices, this tube, which is very heavy, CPU, monitor, keyboard, whatever. This is very heavy, much bigger. And of course, that also results in the level of advancement. Is that a correct word? This is much more advanced, like Mr. Ethan said. Okay. If you or I use a light microscope just like this, how much time would an object be able to be magnified? It could magnify objects as many as 1,000 times. 1,000 times, okay? 1,000 times for some microscope. Even though for some microscope which use ocular lens like 16 times, there is 16 times we have at school, it will be 1,600 times, okay? 1,600 times bigger, okay? But this one, the electron microscope, able to magnify objects as many as millions of times. Awesome. So awesome. Do we have it? No, it's way too expensive. Okay, it's too expensive. Do we need it? No, not yet. In secondary school, we don't really need it. Okay, so to give you a what is that? Quick look on the difference, the capabilities of each. I'm sharing with you this video. Okay, I mean you can browse this in your browser on YouTube. Okay, so this is the first video. Mr. Cover. There he is.
Welcome to Lightbulb Biology. I'm Mr. Corver, and today we are talking about the microscope.
Okay, I'm going to skip a lot of parts of this microscope. Well, not microscope, this video. I mean, sorry, this video. Because what I want to show you is that Mr. Cover here shows us about how a microscope works, okay? And then he also shows us what we can see under a light microscope, okay? See, I'm going to skip some parts, okay? Like, this is a video, okay?
I can't see anything. Use your open eye, Frank.
That was funny for me. I mean, that man over there just now, he got one eye closed, okay, one open, and he tried to look into the microscope using the closed eye. Okay, that was funny for me. Okay, but Mr. Cover here show us something. He used this.
It slips over the eyepiece and allows me to attach my phone to it to record what's going on my
I really
slides.
hope I can have
I
that.
highly recommend it.
tool.
I'll put a link in the description if you want to check it out. I bought mine off of eBay for $30, but it's also being
What?
able to.
$30? eBay? Oh my gosh, $30. I want to have that kind of thing, yeah, to observe things under microscope using mobile phone. But $30, not to mention the shipping. Oh my gosh.
Purchased on
Amazon.
Amazon. So with that, we are going to also need a PDF. There is a file that is linked below that
Never
is the
mind.
link for you
I'll
to download
skip it.
free.
Okay.
To the microscope. I love using the cell phone as an introduction to the microscope. Placing the screen under there really gives some cool images of what a phone is actually made of. Here we're looking at an old phone and a new phone, and you can see the evolution of the screen. So in the next couple slides, we'll now look at how stuff moves underneath the microscope. Here I'm scrolling down and you can see the image moves up. Here I scroll up and the image moves down. Here I scroll to the left and the image moves right. And here I scroll to the right and the image moves to the left.
I hope you paid attention on that just now. What we can conclude is that the image that you see under a microscope is inverted. When he scrolled down, the image moves up. When he scrolled up, the image moves down. When he scrolls right, the image moves to the left, and so on and so forth. See, you always have to remember that the image under a microscope is inverted. Okay, guys? Later in grade 8, you are observing, for example, the circulatory system of a fish. Circulatory system of a fish means the blood system. You will be able to see later the flowing blood inside the body, inside the blood vessels of a fish. If you then see that the blood is moving towards the right hand side, and you see the beginning of the tail of the fish on the right, means the location of the tail of the fish is on the left, inverted. Remember that, okay? Remember that. The image you see, you observe under a microscope is inverted. Okay, carry on. Yeah, inverted. I'm continuing. Now, he used mobile phone to show how amazing microscope works. Okay, but he also go to the wild and he took a sample
Very
from
bottom,
this
and placing that under the microscope.
nasty water, he said.
Because there are some really neat things that live in nasty
water. Okay.
So we're going to use the Tpuc method. Number 1, turn your microscope on. You would be surprised at how many people forget to do that and wonder why they can't see anything.
Really? Seriously? Forgot to turn on the
Next step, make sure you select the correct power. I always want the lowest power that the microscope can handle.
Okay, guys, what he wants is something we strongly recommend you to do. Because when you are observing things using the microscope, you will have several options of objective lenses to be used. Okay, these are the objective lens located inside one part called nose piece. It will be explained later. But it got a ring on it. Yeah, yellow, red, blue, and white. Okay. The length of the nose piece, nose itu hidung, piece itu potongan hidung. Serem juga ya. Okay, the length are different. Okay, the red is the shortest one, and then yellow is the second shortest, and then blue is the second longest, and white is the longest, something like that. But it also indicate the power of magnification. How many times can or will an object be looked, be appear compared to its original size? The red one magnify objects as many as 4 times, 4 times bigger. Okay, the yellow one 10 times bigger, blue one 40 times bigger, the white one 100 times bigger. I hope you can follow me. Is there any question? What we strongly recommend you to do, what Mr. Cover just now said, whenever we are observing things under a microscope, always start with the lowest power of magnification. You will always start with the one with red ring, which is 4 times. Do you understand that, everyone? Can you open up your your microphone and tell me whether you can follow me or not? Can you follow me, everyone?
Yes.
Yes.
Oh, thank God. Thank God. Thank God. I mean, displaying my desktop prevent me from being able to see the video of yours. Okay. So again, whenever you are observing things under a microscope, you better be using the lowest power of magnification objective lens, which is the 4 times. This is important. Okay. Well, later in the lab, when we go to school together, you will use the microscope. Okay, and then it will be fun. Okay, carry on.
Because it allows me to find my specimen easier.
See, blue, that's 40.
And it also won't dirty up a lens. As you can see here, this light actually contacts the highest power we have here, and that lens can get kind of
He said the highest power means on his microscope, there is no 100 times. Okay, so there, the highest is 40.
So I'm going to go to our lowest power, which in this case is
That's
red,
the lowest.
4x. I will then...
Okay, you in your spare time, please browse this kind of videos as well, okay? Instead of just playing games, online games and so on. I mean, this will be more beneficial for all of you. Okay, I'm going to continue what we have here. Okay, this is what we see. Ah, cute. Maybe what you are about to see now could be considered as cute for some, but could be considered as gross for others. Okay, so let's see.
So that is the TPUC. Turn on, power, up the stage, center the slide.
This is what you can see using the light microscope, using a light microscope. See? Cute, right? No, it's gross. It's cute. It's gross. It's cute. It's gross. Okay? Magnify. Higher. Focusing. There. Is it still cute? Relative.
I'm getting even more disturbing, disgusting.
Yeah, of course. Understand. I understand that. Yeah, like I said, for some of you, this could be disgusting. Okay, but I hope, guys, that this is also the
two microscope images.
I'm going to. Okay, I guess that's it.
That's the microscope.
At least you watched how object would be would appear under a light microscope. Okay, now I'm going to show you another video, the next video, which will show you images we can observe using electron microscope, and we will prove Mr. Ethan's what Mr. Ethan said just now about the electron microscope is much more advanced. Okay, let's see. Yeah. Amazing.
There is an incredible hidden miniature world around us unseen to our naked eyes, and science has made it possible for us to enlarge, look and capture the amazing details of this micro world. When viewed under an electron microscope, even ordinary things look so weird
I
that
think
they
the guy
seem to
is
belong
Indian
to a
or
parallel
something,
universe because they can
not native due to the accent. Okay, but we will see very soon.
magnify an object by thousands or even millions of
Ah.
times with incredible things that surround us.
Guys, what you
Some bats have
see on your screen is a bat tongue. Lidah kelelawar. Tah jika kieu tingali. Astaga enggak tuh. Ya, ini lidah kelelawar nih, bat tongue. Ya, bukan lidah tempat tidur, ini lidah kelelawar. Ya, luar biasa ya. Lanjut.
like structures on the tip of their tongues,
Ya, jadi kalau
which
dia
help them slurp
ngisep
nectar
madu,
far more
bisa
efficiently
keambil
than
banyak
a
nih kayak
smooth
gayung mikro gayung ya, gayung mikro yang banyak sekali ya, luar biasa ya lidah kelelawar. Ini jarum hipodermik dengan darah. We can see these red parts. This is obviously an erythrocyte. Okay, an erythrocyte, sel darah merah. And a group of erythrocytes, RBC, red blood cells. Okay. Nah, stigma of a sunflower, kepala putik dari sebuah bunga matahari. You can see how awesome the image can be obtained by using electron microscope. Oh yeah, just to FYI, okay, there are two kinds of electron microscope. First, TEM. Second, SEM. TEM is transmission electron microscope. SEM is scanning electron microscope. Both are able to magnify objects as many as millions of times. Okay, like 1 million or something like that. But the difference between the two, SEM and TEM, is that the TEM produces two-dimensional images, more or less like that. While the SEM, scanning electron microscope, will produce an image like this, what you see on your screen. This is an SEM, definitely. Okay, SEM. Just now the yes.
Is it weird if I couldn't see the size of like I couldn't see the thing that a microscope can see, but I can feel the micro things. I can feel it, but I cannot see it.
What do you mean you cannot see it? Where?
So about the the like if we look at the microscope, right, there will be small things that we couldn't see by our naked eyes. But for me, I can feel them.
Wow. That's who's this? Sorry.
Hagios.
Okay, Hagios. Oh yeah. That's interesting, Mr. Hagios. But can you like describe what do you mean by feel?
So let's take an example of the sunflower. We can see those little strokes there in the middle.
Yes.
And if I touch that, I will feel something that goes pretty much long, and it feels soft and have that texture of I couldn't describe it by words. It's just described by feeling.
Wow. Yeah, for me, what you've just described to us is something interesting, Mr. Hagios. I mean, you can see this in different way, in the sense of awesome way. I mean, feeling that you've involved more than just your sight and others. That's awesome, Mr. Hagios, and I guess that will help you in understanding this kind of things better than others. That's awesome, Mr. Hagios. Yeah, interesting. I myself, looking at this image, it's to me is so awesome. Yeah, it's so awesome. I mean, wow. Yeah, thank you, Mr. Hagios, for setting that. That's awesome. Okay, anybody else wanna say something or share something with us? Okay. More and more. Tar- this is an interesting creature, everyone. This one, tardigrade.
This
Water
is a
bears.
tardigrade, also known as water bears. They are one of nature's most interesting animals. They are among the most resilient animals found on Earth and are almost
It is able to live
indestructible.
in a very hot temperature. Oh, okay. Is this cute?
This
Does
is
this
a
look cute
silkworm.
for you guys? Ulat sutra. Ini ulat sutra, guys. Tampak tampan, cantik, ganteng. Oh my gosh.
Like
Silkworm caterpillar. This is
Something
but
that out
this
of a nightmare.
okay. So I hope that none of you would dream about silkworm caterpillar later at tonight. Okay. But what you need to see here is that how amazing the detail can be obtained by using electron microscope. Okay. I'm not going to continue showing you these. Okay, another worm. Okay, another worm. Okay. Wow. Okay. Okay. This is at the head of an insect, I guess, a bug or something. And you can imagine if this thing is as big as Godzilla, that will be scary. Yeah. Even to see the hair it's alone. Oh my gosh, so scary. Okay. Interesting, guys. So I hope you can see how interesting observing objects using microscope is, and pray to God that we could go to the lab and observe things using our microscope. Okay, even though we only have the light microscopes, but this is awesome. So browse your internet for something useful like this. Okay, I'm going to stop using this video. Okay, I'm going to continue to our main objective, which is we only have 17 minutes, 16 minutes left. My bad. So actually what you are doing now is you are going to have a worksheet, and you fill up the worksheet by referring to the video I'm going to share with you. Okay, so here comes the link to the worksheet, and I don't think you can click on your screen to get the what is that, the link. Okay, to go to the link. What have I done? Just go to, okay, bit.ly, this one. Go to here, Command C. I'm going back to the here. I'm going to attach the link in the chat. Hopefully it's there. Oh, it's not there. bit.ly/LKS microscope. Oh no, bit.ly/LKS microscope pakai bahasa Indonesia, like that. Okay, go ahead, get the file, everyone. Get the file. Don't bother asking or requesting permission to edit the file in the cloud. No, just as long as you can access the file. Yeah, Mr. Hagios, the one on the screen is not a link. Even though in the PowerPoint file I made, this is a link, but it only applies to me. A link for me. So what you need to do is to go to the your browser and bit titik li spasi LKS Microscope. Coba berhasil tidak? Kok nggak berhasil ya?
Bisa, Mister. Bisa.
Oh bisa ya? Oh ya. Thank God. Because it's in the chat it's just like that. Yes.
Unless request edit access.
But can you just download the doc file?
Yeah.
Can you just download it? Because before in the previous class, got the same. The student got also the same. I mean, it's like he had to request edit access, but as far as you can download the file, that's enough. That will be enough. Please don't sing "Never Be Enough." Don't sing that, please. Never be enough. Yeah. Any problem, everyone? Got it? Everyone? Got it?
Student, give a response to your teacher. Do you have any problem or you already
No, no.
Anyone?
No, sir.
Any problem? Okay. Hopefully there is no problem. If there is a problem, you can immediately talk to me, except if the problem is financial problem. I cannot help you with that. Okay. With the assumption that all of you already gotten the... Oh, wait. I can also try to share the file using the chat. Why didn't I do that in the first place? Wait, please. I'm trying to share the file. I'm trying to share my file. La. Here it comes. Hopefully it can. 112 kilobyte. Oh, bisa ternyata. Sorry guys. I hope it would be arriving very... Yeah, sent successfully. It's in the chat, everyone. It's in the chat. Worksheet Microscope 2021. Go ahead. If you got problem in using the link, you can just download the file in the Zoom chat box. Okay. I have to go then. You will fill up the worksheet referring to the video, and you can also, of course, using other what is that, sources. So I'm showing you something else now, which is this one. Okay, which is this one. Guys, can you see the worksheet file on my screen?
Yes, sir.
Oh, thank God. So this is the worksheet. Okay, you will fill the blank labels here with the correct label, of course. Now below you will see a table, and you see there are excluded the header. There are 14 rows. Okay, 14 means this should be related to this one. Means what you fill up in this one, number one, should also be the what you fill in like this. Yeah, number one. Then it should be also the one typed here, number one. If number two is revolver, okay, number two is revolver, then here number two should be revolver, something like that. Okay, that's how it's going to work. And I'm going to go back to the PowerPoint. Now, here's the video. You can access this video on my YouTube channel, okay? YouTube channel Biomolecule. Hi everyone. Today we are going to learn about microscope, video, which is exactly this one, but without the subtitle. So you can choose any which one you want. Okay, the one with subtitle and the one without subtitle. We are going to learn about the parts of a microscope and their functions. We use microscope to magnify small objects so that they can be observed and studied. I'll start from the bottom. First, the base. the bottom of a microscope used for support. Then we got the light source. This could be a mirror or a lamp. The light will be directed through the diaphragm, which will control how much light can pass through and illuminate the object, okay, so that it can be observed. Three parts, which are the base, kaki microscope. Okay, if you see the worksheet again, where is the worksheet? Here, guys. Specification means the function. Okay, so number 14 can be filled up. Number 14 is the base. Okay, and then the function is to support the microscope, something like that. Okay, microscope, something like that. Okay, that's what specification means. So if it is not the function, then it's specification. For example, the objective lens to 20 times, I know 40 times, 10 times, 4 times, and so on and so forth. Okay, go back to the video. And then we got the light source and the diaphragm. Okay, diaphragm here. Diaphragm controls how much light able to enter and pass through the object, so the object can be observed. Okay, diaphragm is over here. We can control the opening using this lever over here. Not really able, clear, but it's here. If you turn this, it will open and close the diaphragm. Okay, and next. Then we have the states. It is where we put the slides, the object that we observe. The slides are held by states clips to prevent it from being removed. When we are observing an object, we can select the objective lens we are using by turning or rotating the revolver. Objective lenses are located in the nose piece. The commonly used objective lenses are 4 times, 10 times, 40 times, and 100 times. States, meja preparat, stage clips, penjepit preparat, supaya nggak kemana-mana. Lalu ada revolver itu dipakai untuk memilih. Okay, it can be turned to select which nose piece you are going to use. Okay, means selecting which magnification you are going to start with. Okay, and this is what we call the nose piece, nose hidung, piece potongan, nose piece potongan hidung. Serem banget ya. Nah, actually the objective lens is a lens located inside a nose piece, guys. So nose piece is like a house of an objective lens. Okay, so the nose piece with this red ring is has the 4 times magnification of objective lens. The one which is yellow is 10 times, blue is 40 times. So nose pieces with objective lens in it. Why do we call it objective lens? Because the lens located near the object. Okay, and on the other hand, there is another lens here located near the eye, a lens located near the eye, a lens located near the oculus called the ocular lens. Objective lens, ocular lens. Objective lens, ocular lens, nose piece, eyepiece. Carry on. Objective lens is connected to another lens called ocular lens, which is located in an eyepiece. The tube that connects the two is called tube. When you are observing things using a microscope, very likely that you have to do some focusing to get good, sharp, clear, and focused images. You don't want blurry images, do you? For this purpose, we are going to use first the coarse focus knob. Some other books call it coarse adjustment knob. Okay. You turn this knob, the tube will go up and down. Be careful. You don't want to break the slide. If you turn the coarse adjustment or coarse focus knob, and you don't you're not careful enough, the nose piece could like press down the slide and break it. Okay. So you have to see from the side if you are observing objects. Second, if you can already find the sharpest image using the coarse focus, but it's not satisfactory, it's not sharp enough, then you can use the fine focus knob. This will refine your image even sharper. Well, I guess those are the parts of a microscope. Hold the arm firmly if you want to carry your microscope somewhere else, okay? Support the microscope with your other hand so that the microscope wouldn't fall and broken. They're not cheap, by the way. Thank you for watching this video. May God bless you all.
Be strong.
Okay, that was the narcissistic part of the video, and thank you everyone. Is there any question? Oh, Clarence sudah ada ya. Is there any question? If you got no question, then go ahead, fill up the worksheet, complete it, and then there will be an assignment pouch on Scholar for you to submit the worksheet. Along the way, you got question, never hesitate to contact me. And I guess that's all for today. Stay healthy, stay safe, and stay happy. That's important. You may leave. Goodbye, everyone. Goodbye, everybody.
Thank you, sir.
Everyone.
Thank you, sir.
Bye.